Good afternoon, everyone. I'm Sir Franz, and this is our first lesson in anatomy and physiology of the different organ systems of the body. So, we will be discussing for this lecture the blood. Relevant chapters that you can use to study this topic would be your main textbook of Marieb, chapter 10. And I also took some information and pictures from our additional reference, Guyton and Hall. So let's start with our discussion with a summary of quick facts. Hematology is the branch of science and medicine concerned with the study of blood. So if we are going to break the word down, hemato means blood and logy means the study of. Okay? Blood is a tissue, not an organ. You have to understand later in our lecture that blood is composed of multiple cells coming together and being suspended in plasma. Compare that to an organ, which is made up of different tissues. So if we define a tissue again, it is a structural organization of the body made up of different cells. And so blood will fulfill that definition. Blood is formed in the bone marrow, and blood is a means of distribution in the body. This means that blood carries with it oxygen and nutrients from the gastrointestinal tract and the heart and delivers it to the peripheral cells of the body. And at the same time, the waste products of the peripheral cells, carbon dioxide, and other metabolic wastes, go into the blood. And from the blood is given into the kidneys and the liver for elimination. So, if you think about it, blood is like a highway where you have different branch points and cars from different locations enter and exit. This picture tells us the composition of blood. So we start with this picture of an arm and phlebotomy is done, which means we puncture a vein using a syringe. So to make a hole, that is the term tomy, to make a hole. And phlebo means vein. So phlebotomy means to make a hole in the vein for the purpose of taking out blood. So if we take out blood and place it in a test tube and place the test tube in a centrifuge, the centrifuge will spin the test tubes around and there will be centrifugal force that will push the components of the blood into the periphery or into the distal portions of the tube. This will force the blood to separate into different layers based on the weight of the component. So kung gaano ka bigat yung component ng blood, yun yung magde-determine kung nasan sila after the centrifuge. So you will observe here that after centrifuging, the red portion, which is made up of red blood cells, very heavy because they are filled with hemoglobin, takes up the most inferior portion of the tube. Ang pinakamagaan would be plasma, made up of water and dissolved substances. It will take up yung superior portion ng tube. And what would be in the middle would be the buffy layer or buffy coat, which is made up of white blood cells. So itong puti na nasa gitna, this would be your buffy coat. So again, after centrifuging, yung pinaka nasa ilalim would be red blood cells. And then, more superior would be the buffy coat made up of white blood cells. And superior to that would be plasma, made up of water and the dissolved substances. So, if we are going to take the total volume of this tube and compare yung proportions occupied by red blood cells and white blood cells and plasma, we will have the following percentage. Okay? So, yung plasma, the water component, tapos mayroong nakadissolved na substances, will take up 55%. So, anong ibig sabihin nun? Kung kukuha tayo ng 100 ml ng blood at sinentrifuge natin, 55 ml of that would be made up of plasma. And 45 ml of that 100 ml of blood will be made up of formed elements, otherwise known as cells. So let's take a closer look at plasma. So plasma is basically a solution, no? Yung solvent natin doon would be water. And the solutes would be salts or electrolytes, plasma proteins. These are proteins made by the liver and excreted into the plasma. Okay? So, galing siya sa liver. And substances transported by blood. So, ito yung other solutes natin. Like nutrients, waste products, respiratory gases, even oxygen and carbon dioxide, and hormones. So, this will be what you will find in the plasma. Ito, in total, would be the formed elements made up of red blood cells and white blood cells. Sabi ko nga kanina, yung Duffy coat or Duffy layer. So, maraming klase ng cells or blood cells na nandito. Majority of that would be erythrocytes or red blood cells. So, cyte means cell and erythro means red. So if you combine the two words, erythrocyte, that would mean a red blood cell. The function of that would be to transport oxygen and carbon dioxide. So from the lungs, the red blood cells will take up oxygen, and as it is circulated in the blood, passing through the different blood vessels and capillaries, it will release that oxygen into the peripheral tissue. And likewise, doon sa peripheral tissue, where metabolism occurs, carbon dioxide is continuously produced. And that carbon dioxide from the peripheral cells will then go into the red blood cells. And the red blood cells, as it circulates, will then reach the lungs, where it will let go of carbon dioxide and take up oxygen. You have to take note that in the laboratory tests that we have for blood, for example, a complete blood count, it will tell us hematocrit, how many percent. Hematocrit means how much of the volume of blood is taken up by red blood cells. Okay? So a hematocrit of, let us say, 35% means if we get 100 ml of blood, 35 ml of that blood, the space is occupied by red blood cells. The other component of blood, the cellular component of blood, is leukocytes, no? Or are leukocytes. Leuko means white and cytes mean cells. So these are your white blood cells. And the primary function of your white blood cells is for defense against microbes that have invaded the body and immunity. Okay? So we have different types of white blood cells: neutrophils, basophils, and eosinophils. These three together, we call them granulocytes because inside their cytoplasm, yan, makikita nyo may mga tuldok, there are granules. That's why we call them granulocytes. And lymphocytes and monocytes are white blood cells which do not contain granules. Kaya ang tawag natin sa kanila ay agranulocyte. Okay? And the last component, formed element of the blood, would be platelets. But you have to understand that platelets are not cells themselves. Kasi wala na silang nucleus and wala na silang organelles, no? What they have are granules. So, etong platelets, you will understand as platelets develop, yung platelets na nasa peripheral blood, yung kinuha natin, would be fragments of a larger cell. Okay? Kaya ang platelets are not actually cells, but just portions pinched off from a bigger cell. Okay? And the function of platelets would be for blood clotting. If we take a section under the microscope, so we place a drop of blood on a glass slide and then smear the blood with a cover slip or another glass slide and look at it under the microscope, this is what we will see. Okay? So, etong brown structures here are actually red blood cells. Okay? But we do not per se see the red blood cells because of their lack of structure, no, that binds to dye. So yung pangpakulay, hindi dumidikit sa kanila masyado. Kaya they will look like this. So these are your red blood cells or erythrocytes. Ayan sila. And those with distinct nuclei would be white blood cells. So this is a lymphocyte. These are neutrophils. Okay? So right here, you will understand that red blood cells do not contain a nucleus. And why is that so? Red blood cells during their development will let go of all their organelles, including the nucleus, so that it will have more space for hemoglobin. Okay? And the small dots here, these are platelets. Okay. So this is again an image of a blood smear under the microscope. Let's now go into the individual cellular elements of blood. So we first describe here in the picture a red blood cell. So a red blood cell is round or ovoid, oval shape, and it has a biconcave disc or structure. So merong carved out portion in the middle on one side, and on the other side there is another carved out portion. So what is the reason for this? This carving out of the middle would increase the surface area of one red blood cell. If this will be even, no, kung wala siyang depression in the middle, the surface area of the red blood cell would be lower, and therefore its ability to exchange gases will decrease. Okay, so this carved out portion, the biconcave structure of the red blood cell, increases its efficiency of carrying oxygen and carbon dioxide. You will also notice that this red blood cell is colored red, and that red coloration is because of the red pigment known as hemoglobin. Okay? Hemoglobin has a central iron atom. So sa gitna ng hemoglobin structure, which you will learn more in your biochemistry, you will find the element iron. And iron, like rust, will color the red blood cell red to red-orange. Okay? So another unique aspect of the red blood cell is that, again, there is no organelle inside. And the reason why red blood cells push out the organelle is to make space for hemoglobin so that it can carry the maximum hemoglobin it can. And the purpose of that is to take up oxygen and release carbon dioxide. So I described a while ago that as red blood cells develop, they will push out the organelles from their interior. So if we take a blood test, let's say a complete blood count, and we see reticulocytes, that would mean those are red blood cells which still have endoplasmic reticulum. Ibig sabihin nun, hindi pa nakokompleto yung process ng pag-release ng red blood cell ng kanyang organelles. Therefore, a reticulocyte, a red blood cell with an endoplasmic reticulum, is an immature red blood cell. So, ibig sabihin na dumadami yung reticulocyte, the bone marrow is producing a lot of new red blood cells. So here I'm showing you the image of how the bone marrow regulates the amount of red blood cells in the blood. So number one, we start with the stimulus. And that stimulus is low blood oxygen carrying ability. So what triggers the bone marrow to produce red blood cells? That would be low oxygen in the blood. If there is low oxygen in the blood, then the bone marrow must compensate by making red blood cells so that the blood can carry more oxygen again. So how does that happen? The low blood oxygen can be a result of a decreased number of red blood cells. It can be the result of decreased amount of hemoglobin, which means the number of red blood cells in the blood remains normal, but inside each red blood cell, the hemoglobin goes down. No? So kahit normal yung dami ng cells, red blood cells, kung konti lang yung dala-dala nilang hemoglobin, then that would mean low oxygen carrying capacity also. Or the air that is being breathed by the patient has low levels of oxygen. For example, when you go or live for a long period of time high up in the mountains, where the oxygen level is lower, no, due to altitude. So, what detects low blood oxygen levels? That would be the kidneys. So the kidneys will be the first one to detect that the blood is poor in oxygen. And what will be the response of the kidneys? It will release this hormone. This hormone is called erythropoietin. Poiesis, which is the source of the term poietin, poiesis, means the synthesis of new blood cells. So erythropoiesis would be the synthesis of new red blood cells. And the hormone that facilitates the synthesis of new red blood cells would be erythropoietin. Okay? Again, let's go back. The kidney senses that low oxygen levels are present. And so it will release erythropoietin into the blood. And what will happen? The erythropoietin that is in the blood circulates throughout the body and it goes into the bone marrow. No? Yung bulalo ng ating buto where red blood cells and other blood cells are formed. The erythropoietin that goes into the bone marrow is utilized by stem cells to hasten up the development of red blood cells. And so you will have enhanced erythropoiesis. And this increases the red blood cell count and the hemoglobin concentration. And the final effect of that would be an increase in oxygen carrying ability of the blood, which will normalize the stimulus. Here I'm showing you how red blood cells develop. So, my goal in this slide is just to show you that as red blood cells develop, first they are They still have organelles, and they look like other normal cells in the bone marrow. But as they develop, they will continuously lose these organelles. Yan. So makikita mo pakonti ng pakonti at paliit ng paliit yung size, and eventually it will develop the biconcave shape. Okay? And what is the goal again? To increase the amount of hemoglobin inside the cytoplasm. So the development of red blood cells occurs as in this manner: proerythroblast, basophilic erythroblast, polychromatophil erythroblast, orthochromatic erythroblast, then reticulocyte. Ito yung sinabi ko kanina, that can be seen in the blood and is a marker of increased red blood cell synthesis by the bone marrow. And finally, we'll have the mature erythrocytes. The picture here on the right shows us what the blood cells, the red blood cells, how the red blood cells would look like if you have different diseases. So, for example, if you have the disease microcytic hypochromic anemia, that would mean that the red blood cells are small-celled, hypochromic, decreased coloration. So the red blood cells are small and they are pale. So that is your microcytic hypochromic anemia. This is how your sickle cell anemia will look like, and this is due to a genetic defect. This is how your megaloblastic anemia would look like. Megalo means large, no? And they are blast-like, which means they are immature looking. So they are large and they are immature. And that is due to the failure of the nucleus to develop. And why is the nucleus failing to develop? Because of a lack of vitamin B9 and B12, no? Folic acid and cyanocobalamin, which are essential for nuclear maturation. Next, let's go to white blood cells. White blood cells come from one stem cell parent, and this is your multipotential hematopoietic stem cell. Ibig sabihin nun, ng multipotential, marami siyang pwedeng maging products. No? It can develop into a lot of different types of cells. Okay? So, let's now go into how the white blood cells develop. This multipotential hematopoietic stem cell can become a myeloid progenitor or a lymphoid progenitor. Myeloid means in the bone marrow. So this cell is in the bone marrow. The common lymphoid progenitor is still in the bone marrow, but as the cells mature and differentiate, as they specialize, these cells will exit the bone marrow and go into the lymph nodes. Okay, kaya siya lymphoid. But the two are still in the bone marrow. The lymphoid progenitor, as it develops, again, will exit the bone marrow and go into the peripheral lymphoid organs, such as the lymph node, the spleen, and the thymus. So, your myeloid progenitor will then be able to make different types of cells. And the tendency of the myeloid progenitor to become a particular type of cell would rely on the amount of hormones or interleukins present in the blood. For example, if in the blood there is a lot of erythropoietin, that will cause the myeloid progenitor to become an erythrocyte. Okay? And so, by different hormones, makokontrol ng inyong bone marrow ang pagdevelop ng myeloid progenitor. The myeloid progenitor can become a megakaryocyte which is a very big cell with lots of nucleus, which pinches off its cell membrane and cytoplasm directly into the blood to form platelets. So thrombocytes are platelets. Your common myeloid progenitor can become a mast cell, which is important for allergy, okay? Because it contains histamine. Your myeloblast is the common source of the different granulocytes, and a granulocyte, the monocyte. So your myeloblast can produce a basophil, a neutrophil, an eosinophil, and a monocyte. No? So, isa lang yung pinanggalingan ng apat na ito. And the monocyte can further differentiate, no? It can further specialize into a macrophage, which is a very effective cell for phagocytosis and destruction of bacterial cells, and dendritic cells, which help the immune system form memory, no, or specific defenses against a pathogen. Your common lymphoid progenitor can become a natural killer cell, which is important for killing virus-infected cells and cancer cells, or small lymphocyte cells, which can become a T lymphocyte or a B lymphocyte. Your T lymphocyte is important for killing virus-infected cells, while your B lymphocytes will further develop into a plasma cell which will secrete antibodies. So in this table, summarized yung different white blood cells. The normal number of white blood cells would be up to 11,000. Okay? We have different types of white blood cells, but the major classification would depend on the presence of granules in the cytoplasm. So, yung mga tuldok-tuldok. So, ayan. Okay? If they have granules, we call them granulocytes. So, we have three types of granulocytes: neutrophils, eosinophils, and basophils. Neutrophils are the most important white blood cell for acute infection. So they are the ones to go into the site of infection the fastest, no? And they are active phagocytes, which means they will engulf a lot of pathogens and dead cells. Okay? And they are, again, the first to enter the site of infection. Eosinophils are also granulocytes, but they specifically attack worms. So for example, if you have intestinal parasitism and you take out a portion of the intestine where the parasite is found, and you look at that intestine under the microscope, you will see a lot of eosinophils. And the eosinophils are there because they specifically attach to parasites and digest the parasite piece by piece, okay, by secreting digestive enzymes. Eosinophils will also increase in the blood whenever you have allergies. So, for example, if you have an asthma attack, or if you have anaphylaxis, okay, those are severe allergic attacks, then you will see an increase in eosinophils. Basophils are granulocytes that contain histamine, and the effect of histamine would be to cause vasodilation. Okay? Okay. The second type of white blood cells do not contain granules, and they are agranulocytes. We have agranulocytes, two types, the lymphocyte, and the lymphocyte is a specific cell which has a particular function of producing adaptive immunity. So adaptive immunity is a type of immunity that is very specific. For example, the pathogen or the microbe that infects you would be herpes simplex virus, HSV. Then there will be a particular lymphocyte specific only for herpes simplex virus. Okay? So, if you have other pathogens infecting you, there is a specific lymphocyte for that. No? Compare that to the actions of the granulocytes and monocytes, which is non-specific. No? So, if there is a pathogen that enters, whatever that pathogen is, the granulocytes and monocytes will react to that. No? Wala silang pinipili. But lymphocytes, there are specific lymphocytes for each specific infection. Okay? Monocytes will further differentiate into macrophages once they are in the peripheral tissues. So the function of monocytes would be to coordinate the inflammation, no? So it will coordinate how the body responds to an infection. So it is a very effective um, phagocytic cell, no? So magaling siyang kumain ng pathogens and dead cells. That's why for long-term infection, it serves as the cleanup team. Okay? And it helps lymphocytes by telling to lymphocytes what particular pathogens are present. No? So it is how the lymphocytes know what pathogen is present through the action of macrophages. So, kumbaga, the macrophages are in a mall, sila yung marites, no, na magsasabi na may kaguluhang nangyayari. And the lymphocytes would be the security guard that will go into the site of commotion. So, para siyang gano'n. This is how white blood cells exit the blood. So first, there will be signals released by the injured tissue or the pathogen. And these chemical substances or chemokines would increase in concentration as long as the pathogen is there. So you will have a concentration gradient. Mas marami yung chemokine dito, konti lang sa blood. So the effect of that would be to increase the permeability of the post-capillary venule or the capillaries. So yung mga maliliit na blood vessels, they will have increased permeability. And the lining of the blood vessel will be very sticky because of this substance, no? The chemotactic substance released. So once it becomes sticky, as white blood cells travel the blood vessel, they will encounter the lining of the blood vessel, which is very sticky, and so they will stick into the lining. This is what we call margination. And the white blood cells, after sticking to the lining, will become very fluid. Magiging parang amoeba sila, no? They will be able to fit and change their shape to exit the blood vessel and go into the peripheral tissues where the infection is present. So the changing of the shape and the fitting, no, of that shape to pass through the blood vessel is what we call diapedesis. So they will reach the site of infection and perform their function, which is to kill the pathogen and engulf the pathogen and engulf the destroyed tissues. So this is how that engulfing process occurs. Ang tawag natin dito would be phagocytosis. The microbe is this capsule-like shape, and maraming antibodies na nakadikit sa kanya, no? Pinalibutan siya ng antibodies. So if this is the macrophage or the neutrophil, which are phagocytes, they will have specific receptors for the antibodies that coated the microbe. The antibodies will bind to the receptors, and it will cause the membrane of the white blood cell to pinch off, no? So, mag-i-invaginate ito, and it will form a phagosome. So, meron ka na ngayong vesicle inside the cell which contains the pathogen. This vesicle will then fuse with another vesicle, but this time this vesicle called the lysosome contains digestive enzymes. So yung bacteria or pathogen magfuse with the digestive enzymes forming the phagolysosome. And the microbe will be digested, and it will fuse again into the cell, releasing the debris. So we have discussed from this point how the red blood cells are unique, what the different types of white blood cells are. Now, let's discuss the function of the last formed element in the blood, which are the platelets. So the primary function of platelets would be to produce hemostasis. So hemostasis, hemo means blood, and stasis means the lack of flow. So whenever there is bleeding, hemostasis will control the bleeding. And that is a primary function of the platelets. So let's begin. Step number 1, you have a blood vessel which has become injured. So nakikita nyo yan? Yan yung sugat ng blood vessel. If this will not be repaired, blood will continuously leak out of the vessel, and you will exsanguinate. And whenever the blood inside the blood vessels, very little na lang, you will have shock and you will die. Okay? So the body has to find a way to prevent you, the patient, from losing blood. And that is through hemostasis. So nasugat na yung blood vessel, blood is continuously leaking. The first step in hemostasis would be to constrict the blood vessel. Okay? So the smooth muscle here in the blood vessel, yan, yung middle layer, will contract. Much like your muscles will contract, the muscles around the blood vessel wall will contract. And as they contract, they will squeeze the blood vessel and decrease the blood flow. So if you have decreased blood flow here, little blood will exit the site of injury. Okay, so the first step is vascular spasm, which will cause vasoconstriction. Step number 2, mapapansin ngayon ng platelets, itong mga puti, that there is a rough surface. So normally kasi, kapag walang injury, the blood vessels are very smooth. But this time, when the platelets attached or went through that particular portion of the injured blood vessel, they encountered a rough surface. And that rough surface is due to collagen sticking out because of the injury. So, what happens? The platelets adhere to the rough surface. They will adhere to the rough surface and they will adhere to one another. So, magdidikit-dikit sila dun sa injured area at magdidikit-dikit sila sa isa't isa. And that is how a platelet plug will form. So, that's step number 2. Injury to the lining of the vessel exposes collagen, and the platelets stick to collagen. The platelets then will stick to one another and at the same time release chemicals, no? That will activate other platelets. So, kumbaga, mag-uusap-usap na naman sila. May isang platelet na naka-encounter ng rough patch here. Magdidikit siya dyan at magre-release siya ng substances like adenosine and thromboxane. I-release niya. And that adenosine and thromboxane will serve as a messenger to other platelets na merong injury doon, so that the other platelets will go there and stick to that surface, blocking the injured site. Okay? So that process of releasing substances and activating other platelets is called platelet activation. Okay? So didikit sila doon and eventually they will seal off the injury. So ayan, kung mako-compare mo dito sa step 1, meron ka pang malaking gewang. Dito, na-seal na ng platelets. Okay? And so the next step, the platelets will allow proteins that are present in the blood to form some sort of cement. So that cement is what we call fibrin. No? So the clotting factors, which are proteins in the plasma, and those that are released by the platelets, interact with calcium to form thrombin. And this thrombin will change the protein, the plasma protein that is in the blood, fibrinogen, into fibrin. And now fibrin is like a glue, no? Initially you have platelets here, but the platelets here are sticking to one another, cell-to-cell contact, no? Yung pagdidikit nila sa isa't isa, cell-to-cell contact lang. So the third step is actually coagulation. And this is how you form glue, which is in the form of fibrin, to cement the area, no? So yung original na cell-to-cell contact lang, babalutin mo ngayon yan ng semento, which is in this case analogous yung semento to fibrin. So platelets lang muna, and then later on fibrin will coat the entire area, and this will now form a very stable clot. No? And then slowly the blood vessel will repair itself, and then the clot is no longer needed because the blood vessel has been repaired, and the clot will be dissolved. So that is how hemostasis occurs. So through this lesson, we have understood the basic functions of blood, what the composition of blood is, and we have taken a closer look at the actual cells that are present in the blood.